All right. Welcome back, everyone, to session 2. Hope you all found your way back into the room all right and got some coffee or food if you needed it. See, Riley there is mentioning he ran out of coffee, so don't run out of coffee. It's going to be a long day. All right. So, I'm Daniel Enking, CEO of Everflux Technologies, and we've got Brian Walksman here, and we've got Chris Trump as our second speaker for today. So, real quick, before I pass it off to Chris, our sponsor, our title sponsor for this whole event, as I mentioned earlier, is AgroQuar, and they make a really high quality cocoa coir, not using all kinds of toxic chemicals and stuff like that, and sourced in a sustainable way. And then our sponsor for the session is Sweet Heal. So just want to remind everybody that if you have questions, feel free to post in the chat, post your questions. We'll mark them as questions, and we will definitely try to get to as many questions as we can at the end of the session here. So with that, I think that's all we need, and I'll pass it off to Chris Trump to get started.
Hey, good morning, guys. Afternoon for some of you, morning for others. It's awesome to be here with you, and really, really honored to be on here with all these other speakers. I love the work of Efren. I've gotten to talk with him over coffee and breakfast, and just a genius. It's great. Leighton, good friend, and somebody I've gotten to cut it up with a lot of times, and it's great nerding out. Chip, also just all these people way smarter than me, so just wonderful being on here. And then I look forward to hearing what James White said, as I gotta watch these back afterwards. So really cool. And then Craig came out and studied with me on the East Coast last year, or 2 years ago now, and just a wealth of knowledge. Him and Leaf are going to speak. So yeah, great to be here talking with amongst peers in the industry, and like I said, a lot people a lot smarter than me, so it's cool. If you don't know who I am, I grew up farming and learned kind of regenerative agriculture and Korean natural farming on in the Big Island of Hawaii. We farm 750 acres of macadamia nuts, and natural farming protocols and kind of what we discovered made a huge impact on our bottom line as farmers. And it's not finalized. It's not. We haven't learned everything there is to learn about using microbes to grow food on our farm, but we came to a place where we got moving forward. And so that knowledge to make those connections and make those improvements on my farm were, by and large, given with a heart full of openness. My teachers, Master Cho Han Yu, his son, Cho Young Sun, who created Jadam, kind of a subset of natural farming. And when I first learned, Master Cho's daughter, Ju Young, was really vital in me getting some of the nuts and bolts. And so the way they've shared it in an open source format is really a heart that I have to, I feel kind of, in Hawaii we say kuleana, privileged responsibility to continue to pass forward. So we try not to make any of this information inaccessible. And I love the grouping here. All this knowledge is just dropped and left to you to play with for a lifetime. So that idea of sovereignty for farmers, being able to create your own inputs, be able to control that or have your own ability to improve the health of your land and crop production and quality yields, etc., is why we're here today. So I hope this translates to that for you, and I plan to end a little early to extend that Q&A time, so that helping make that connection between some of this information and what you're actually going to do with it on your farm, or how what you heard translates to what you're doing. Really important to me. So prepare those questions, write down some notes, and we'll jump into it. One of the things I want to do today more than anything is kind of pose some unanswerable questions, or expand our view of what IMO is, indigenous microorganisms, and what they can mean for you on your farm in real time, real production, for your crops. And today we're talking about cannabis. So I work in a bunch of different industries in agriculture, and love the diversity of that. And we have a lot to talk about just about cannabis, so I'm stoked for that too. Indigenous microorganism. Why do we have that first word there? We talk about microorganisms all the time in this community, which I'm sure you're a part of if you're sitting here listening to us today. You know, as much as I'd rather be in person talking to you, hashing it up, making you smell things, I love making you taste weird stuff. I also appreciate this format that you can be at home in your cozies and really getting a wonderful download during a time when the world's on fire. So tons of empathy for you guys going through this last year and the year to come. And my hope is that these bits of knowledge are something that we can find a great deal of joy in. Enjoying the journey is a core philosophy in natural farming practices, because you as a farmer are actually the most vital part of your farm. Yeah, microbes are important, but the reality is that very old idiom is that the shadow of the farmer makes the grain grow, or the eyes of the farmer makes the calf fat. In cannabis, the shadow of the farmer makes the flower dank, whatever you want to say, but really your presence as an observer on your farm being one of the most vital and really valuable inputs that you can put into your farm. So we'll talk a bit about that, and more than anything, I would like to pose the idea that we have a lot to learn as a scientific community on the interconnectivity of the microbial community and how that affects crop nutrient uptake and yield, quality, production of biochemicals like cannabinoids. We're doing that. We're seeing it. We're playing it out. Really for the last 4 years, I've been able to get direct data and feedback with the cannabis community. And it's been a lot of fun. There's a lot of great farmers having all kinds of new and exciting successes using living soil to grow their crops. And so I would like, if you're wanting to jump on that, wanting to get in that more, I would like this to be a maybe less exactly how to do it, and more like if you're like, well, I don't know if I'm going to jump on that whole train of getting into diversity as the word we throw around over and over again, then I would like the outcome of this talk to be that you're like, well, I can't but do it. So, indigenous. Why indigenous? So right now, today, I am in Colorado. It's actually a nice day today, but it's been freezing cold, 10 degrees at night, and that frigid air and the microbes that thrive in it are blowing through an indoor production facility every single day. And there is a slightly different snapshot of indigenous microbes that thrive in the winter cold. And so partnering with those, with what's alive and functioning in our crops, even though our crop production zone is kept at 75 degrees, 40, 50% humidity, The influx of microbes, yeast and bacteria through the air vents is a real addition or a real input on your farm, and how those play or interact with the foliar microbes that are thriving on your plants and the soil microbes that are thriving in your root zones is a real dynamic that we can learn more about, but also even if we don't know anything about, we can benefit from and partner with. And so natural farming and this idea of an indigenous microorganism is saying, yes, we know some things about microbes in community, a little bit. And to partner with it, we don't necessarily have to have all knowledge about how microbes work in community. And so how do we, as farmers, partner with natural law that we can't fathom, we can't see the bottom of, we don't understand the depths and the complex interconnectivities of this beautiful symphony that is nature? What is it that we can do to come alongside or benefit from how nature works in general, even though any one of you or any one of us might not have complete knowledge on the topic? And I propose that you can definitely benefit from natural law and partner with it in your grow, even if you're not a super nerdy scientist, even if you're somebody that likes the stuff but just doesn't have time to learn what that word means or how these microbes work. So rest assured, you don't have to know everything to jump in and play with what we're talking about today on this session. And I would love to kind of jump into the mind-bending complexity that the microbial world is, so that we can say, oh, as much as we understand, we can relax in the ignorance we have as a scientific community, knowing that though we are ignorant of the whole, we can play with and partner with the things we do understand. And it's kind of humbling, as I think most of the scientists on here would probably agree, as you dig into the incredible complexity of the interconnectivity of the microbiological world, that it makes us feel a little bit small. Yeah, microbes are small, but the vast implications of what microbes do for us in our guts, on our skin, in our hair, on our plants, leaf surfaces, root surfaces, stems, and in the soil. It's wonderful and beautiful. So I am somebody that grew up farming, and have a lot of that as kind of what I've come to realize is intuitive understanding that not everybody has if they haven't farmed for a long time. And that is a learning curve. That is something to overcome if that's not something you have as your background. But it's not an insurmountable learning curve, and it is definitely something that you can jump into and learn as you go. So again, just circling around to indigenous, we capture microbes from thriving soil environments, thriving plant microcosms, or areas where the life is gangbusters or going along really well. The plants in those environments are very healthy and self-maintaining. Nobody's tending to them. And really what we look for in collecting our inoculum for our farms is we look for extremely undisturbed or long time undisturbed areas where nature has had time to strike balance and maintain balance. And so what we're doing is we're not necessarily going in to try and get a microbe out of that system as much as we really are trying to go in and get the diversity and the balance that nature has struck, and with as little tweaking as possible, place that into our ag land, our growing environment. And then you'll hear me say the word diversity, hopefully 100 times before we're done here. And that's because in nature, diversity rules. Diversity is your buffer. So, for example, what is the microbe that prevents powdery mildew from running away on your leaf surface? We don't know. But we have found over the last 4 years that natural farmers with foliar application of indigenous microorganism don't deal with powdery mildew. It's not an issue that they're worried about. And a lot of the germ diseases, bacterial wilt or blights or bud rot, these are less seen, less prone. Your plants are less prone to experience these in a highly diverse microbial environment. So what I've done and we continue to do as natural farmers in cannabis production. I'm not personally a cannabis grower. I have as a kid, but not something I'm spending my time doing. However, the amount of time we're spending talking about it and solving problems in the cannabis community through these dialogues has been abundant, and we're finding some wonderful things. So I'd like to share some of that with you, and more than that, I'd like to hopefully blow your mind on the depth of the implications of playing with microbes in a natural, diverse, functioning soil food web or functioning wheel that's turning out and doing the nutrient cycling in your root zones. When we talk about indigenous microorganisms, we're talking about whatever could potentially be in that indigenous soil environment. So everything from the smallest to the biggest of the microbial community, working in conjunction in nature and seeking to establish that on our growing land. And it is really possible. I had the wonderful pleasure. It's a lot of work. We're only 25% through the grow, the garden I'm at today, or this week here in Colorado. But we went through the soil beds. These are living soil beds. Leighton jumped in and he was involved in creating these beds initially, and another friend of ours. The soil layers and the compost. We used river rock, sand, silt, and clay, compost, sand, silt, and clay, and compost and IMO 4 wood mulch. This soil system, the idea here is that it gains maturity over time. The soil continues to condition, and the microbes in a diverse, thriving system are mining some of those minor minerals from those indigenous mineral resources, which Colorado river rock, sand, silt, and clay from here, and compost made in the area from biomass from here. Not that all of that is vitally important. You can do this in, bringing in materials from elsewhere, but some of these things fall into this category of philosophy of ideal situation or ideal environment for these indigenous microbes to be able to mine latent minerals tied up in rock, sand, silt, and clay, and organic matter, and make those on demand available throughout a plant's growing cycle without you needing to manage it. The thing is, in nature, plants are getting incredible nutrients available to them through that biochemical exchange in their root zones. If you have a functioning soil food web or a functioning microbial community that is both diverse and complete. What do those things mean, diverse and complete? Complete is pretty ambiguous. It's pretty hard to nail down. The idea there being the more diversity from our indigenous environment, the better, because in our growing beds, nature can strike balance like it does in general. It's something that nature does well if given the right environment to do it in. Where the microbes are present, they exist there. So I love that people have jumped on in natural farming with LAB. But as we'll jump into this, I have a PowerPoint for you guys. As we jump into that, we will be looking at how limited we are if we are only getting things off the shelf, if we are only getting whatever has been put in a jug for us to apply to our farmland. We're limiting the potential symphony of what nature can do. I mean, it's a blast. I watched a protozoan flagellate swim around like a psychopath inside a testate amoeba that somehow it had crawled into, a dead testate amoeba, and it's going nuts running around inside there, which is just a blast to watch. But if I'm applying LAB to a cocoa system with organic nutrients, I'm not necessarily going to have any of those guys helping me with my crops. And we want that because those are free farm workers. They work day and night, and they don't ask you for anything other than an environment to grow well in. So a huge and very important thing if you are running a living soil system is to maintain proper water. Okay, I can tell you that on the farm side of what we're doing in living soil systems, proper moisture is 80% of your effort. 80% of what you should be focused on is keeping that moisture right. Whether you ebb and flow, you get it wet and then let it dry out, not too dry, and just maintain that kind of ebb and flow that happens just like a rainstorm comes through and wets everything and you wait for the next one. That's great. But if you are harvesting and between cycles, letting everything dry out and get really bone dry, that one thing is drastically knocked down your overall nutrient cycling potential as you plant those next things in. If you're re-wetting when you plant those next things in and hoping that everything's going to be clicking along just as it was when you took your crops out, you have another thing coming, because that dry out has caused all kinds of calcification that can create some weird lockouts on nutrients and definitely kind of take all your really active microbial wheel or engine that's pumping out nutrients for your farm and interacting and preventing disease and put everything to sleep. And then it takes time for all of that to wake back up. So maintaining moisture, especially if you're in a closed system, not an outdoor system, is vitally important, because what you want to do is you want to maintain environment for the microbes that want to work for you, want to work hard for you, but they need proper environment. Just like worms, you dry it out or get it too wet, the worms leave. That's a macro example of what can happen on a microscopic level if you don't maintain proper moisture. So 80% of your farm side, maybe, just stepping away from, as it crosses over to the microbial side, 80% of what you should be focused on is just that wonderful maintenance of proper moisture. And I know this is as basic as it gets in agriculture, but I have found if I don't say that, I am surprised by the missing of that and the cost that is. So too wet is death to your microbial communities. A swamp will take your diversity down really quick. Too dry can create problems with your microbial communities, maybe less than too wet. Your microbes will go dormant and wake back up as proper moisture comes in. But some of the nutrient cycling, some of the things that can happen with a lockout because some nutrients calcify in different ways in that dry soil environment can really hurt you on yields, production, and even create plant stresses that then cause other problems. So I just wanted to pause on that water topic for just a second. We know how important water is as in general, but just in case that helps somebody with perspective, water is vitally important to maintain. And in a living system, you maintain it at all times, not just when the plants are in the ground. So if you have your cover crop on, you water your cover crops, you treat that soil just like you have, 2 weeks to go before harvest or a month to go before harvest. You want to have that same care in between planting as you have while the plants are in the ground, because you're maintaining your living soil's efficacy or function, so that when you put it in the ground, you're not waiting for a wake up. And then on the 80% of natural farming, it's indigenous microorganism establishment. So, there's all kinds of ferments we talk about in natural farming as plant nutrients. And I love those because you have a great deal of control, meaning the right nutrients at the right time, rather than a big stockpile of nutrients in the soil up front, that then maybe there's too much nitrogen going into flower and there's very little you can do to mitigate or bring that down. By having soil functioning kind of like outdoor soil does, without a bunch of preload of nutrients before you put your plants in, but then having all those nutrients that the plant needs through its life cycle available to apply at the right time as it needs it. This has given cannabis farmers using natural farming a great deal of control to maintain those balances that plants need in their systems, nitrogen, calcium, potassium, manganese or magnesium, these interconnected nutrients that the plant needs to balance within their system. You're able with observation to adjust and properly feed. But that's the 20% of this. If you have your IMO, your soil food web, your microbial engine functioning in nature, functioning in your soil, so much of your plant's nutritional needs, especially the minors, are going to be maintained without you having to run around and maintain them. And it's a really beautiful thing. It is real. It's not too good to be true. It's a wonderful function of how plants interact with living soil environments. So I would like to jump in now and just kind of tee us up for asking the right questions and really being kind of properly humble as we approach the incredible depth of what microbial diversity and the natural world is and how microbes can affect that. So I'm going to jump into a couple studies that have been done. I think both of them are two of them are in Japan and one is in Spain, about microbes and their effects on plant production, and both positive and negative. And one thing, I'm making a few hypotheses and assumptions that need further study. There's a lot of incredible studies done in different industries in agriculture that we can learn from and benefit from. But in reality, eventually, we're really going to need to ask some really important questions about cannabis specifically, and we're queued up to do that. You have so many wonderful people interested in the research. Cannabis is the greatest botany project ever done in the history of man that we know of. There's never been a greater study in botany than the genetics and crosses that we've come up in the cultivars of cannabis over the last since the 70s. And so that's pretty special. And I think on that note, we can learn a ton about agriculture and nature in general through the study of cannabis. So I look forward to that. And barring that, or until we have more of our plant-specific things, I think there's things that happen in nature that would be good to know about, so that we can kind of have the right amount of awe as we go into partnering with nature to grow food or grow medicine, as we do in cannabis production. So what I would like to talk about, and this is to a degree hypothetical or a hypothesis, is the implications of the fungal to bacterial ratio in your root zone, in your soils, and how that affects you in your production and your yield. And my assumption, with just kind of taking the input and observations from some incredibly smart people, a friend in Canada named Reese, he's jumped into it, and he loves studying that fungal to bacterial ratio, and we had a good talk about it. But as we are observing, proper amounts of diverse fungi are the key to the soil and nutrient uptake, yield, and disease prevention in cannabis. So you don't want a highly bacterial system. My current working number is somewhere between 30 and 100 to 1 fungal to bacterial ratio. So that would mean in biomass, for every 30 fungi, there's 1 bacteria. So skewed on the fungal side pretty drastically for cannabis production. And we can talk another time or in some of the Q&A about why, but let's jump in a little bit to how this works in other places in nature and what we can take away for cannabis. So this may be a direct effect, this topic, this thought of fungal to bacterial ratio and diversity in your soil may be a very real number that is affecting you on your farm that you may or may not be aware of. What is your fungal to bacterial ratio in your growing zone? What does that look like? How do we tweak or affect that? And what are some of the implications? What are some of the things that may be happening that we don't know of? So, real quick, just want to look at the nitrogen cycle, because nitrogen N is just a great, broad spectrum, or broad brush. We're just painting nitrogen. Plants need nitrogen. Absolutely. Vegetative growth, plants need nitrogen. Cannabis needs nitrogen. What type of nitrogen is the ideal nitrogen for the cannabis plant? In trees, I'm a tree farmer. By and large, trying to learn everything, but in trees, it is ammonium, organic nitrogen, organic N, which is not really easy to buy from a bag. How does that dynamic of the different forms of nitrogen affect us? So my hypothesis, in my understanding, is that our cannabis plants are going to prefer organic N and ammonium that they maintain in their root zone through some wonderful things. We'll jump in a second. So nitrification in nature is a 2-step oxidation process of ammonium or ammonia to nitrate, catalyzed by 2 ubiquitous bacterial groups. Or ubiquitous is just, they're everywhere. This happens in nature all the time. Nitrogen coming down with our rainwater, which is mostly organic N, is oxidized or changed biochemical process through the use of or through the acts of some bacteria and the enzymes they have to make those chemical changes. What does that mean for us? This is actually the farm I'm at right now. It's kind of a panoramic view. They are not curved beds, but we're going to talk real quick about a study out of Japan looking at Japanese cedar and how they, through their through the life in their leaves or in their phylosphere, how they are controlling what kind of nitrogen they eat by the microbes that they partner with in their bodies and in their environment. So, in other words, a Japanese cedar is taking nitrogen and stopping it at the ammonium, stopping it in ammonium rather than letting it progress into nitrates and nitrites, because that is what it prefers to eat. It does best when it eats ammonium rather than nitrates and nitrites, or even ammonia. And so the plant can eat nitrates and nitrites. Plants can do that, but the preferred food being something that a plant itself buffers and feeds itself by the microbes in its environment, much like we feed ourselves by the gut flora in our gut. Plants do the same thing. And so this study is a lot of fun. I like it. The implications there are that we need to ask the question, what is the preferred nitrogen of the cannabis plant, and how best can we give it what it wants to eat for maximum, time for the farmer, if I have more time, and profits and quality, or production and quality. So microbial nitrification and throughfall of a Japanese cedar associated with archaea from the tree canopy. So a type of microbe that lives in the tree canopy and buffers or chooses how that nitrogen available to it is then converted to usable nitrogen in its root zone. Why is this important? So we're talking about a tree study, but I think in general what we're looking at for the cannabis plant is that we're probably closer into the tree or woody shrub preferences in nature than we are to like a tomato plant. And I know they look similar, but I think that that woody stalk that can develop in a really old cannabis. We keep cannabis pretty young, and soft in our production cycles for maximum profit, right? But if you just let a cannabis plant go and never touch it, and what does that stalk look like? Does it look like a tomato stalk? Not really. It's extremely woody and full of carbon, a lot more like a tree or perennial shrub than it is like some of these more delicate crops that we sometimes associate them with. So trees have a microbial life on their leaves that specifically oxidize ammonia, but almost zero microbes that form nitrite. So these are nitrospira, are those microbes that do that work of nitrification towards nitrites. These microbes oxidize ammonia, making NH3. Then, with a little rainfall, it combines to become ammonium. That's that chemical process there laid out. So the trees in nature choose how to eat for their preference. Trees have the ability to suppress soil nitrification through the release of nitrification inhibitors from plant roots, and is termed biological nitrification inhibition, BNI. And so in the zone around their drip line, so this study's looking at rainfall. Rain is full of nitrogen. The plant catches it, inoculates it with the microbes on its leaves, and drips it down to the root zone, where it is then inhibited from progressing into nitrates and nitrites and kept in an organic N or an ammonium, because that is what those roots prefer to eat. I mean, this is beautiful, right? This plant, this tree creates a symbiotic relationship on its plant tissues to inoculate rain. And so that when it goes down to the root zone, it collaborates with the BNI, or biological nitrification inhibition, to prevent it from going to nitrites and nitrates and keep it in an organic and an ammonium form, because as it eats that, it does best. Nature's incredible. It's a blast to play with. And in my view, as we best partner with what nature already prefers to do, we experience, I don't know if easier is the word, but we experience an ease in getting that quality and that vibrant life that we want, rather than if we're trying to hack the system and come up with our own way of doing it. So that is a study. If you want to look it up, you can, right there, Watanabe et al., Springer Plus 2016. So search that, and you can dig into that study. It's a lot of heady stuff. I tried to pull out some interesting tidbits just to kind of give us perspective on how complex and beautiful nature's processes are. And if we are hacking it, and I say hack, that means, you know, we're providing all the nutrients we think are perfect for our plants. That doesn't mean that we check our nutrient knowledge at the door, or that I'm saying throw out everything you know about feeding your cannabis plants and use rainwater. I'm not so much talking like that, as much as saying there are dynamics at play here that are directly affecting you. And if we can move into partnering with how nature does things, rather than coming up with, the extreme would be a salt program, where we're deciding what the plant gets, and it has no access to anything else because there's no nutrient cycling with microbial life. And really, any given farmer is somewhere in between, wild nature and salts, a salt program. We fall somewhere in between this. How much are we deciding and coming up with how a plant eats, and how much are we partnering with how nature feeds it? Who else likes to work with ammonium? So I put some stars. Obviously, we're talking in that last study about trees, but some of these plants that prefer higher fungal, lower bacterial ratio in their root zones, there is precedent, or there is some degree of understanding that they also are liking or more into organic and ammonium for their primary food source than they are into nitrates and nitrites. And the next study talks a little bit about how plants fall into the food they prefer to eat based on kind of where they are in this fungal to bacterial ratio that we're seeing. So early successional versus these kind of late successional. Late successional, in my understanding, as I've dug into the research of it, are going to prefer ammonium over the nitrates and nitrites. And vice versa, early successional weeds, etc., are going to really struggle to eat ammonium and prefer the nitrates and nitrites. So jumping into the next study, this one's a lot of fun to help kind of give us a visual on how this might be affecting our crop one way or another. Early successional weeds, however, can't handle ammonium. In this study, they studied the influence of nitrogen, of the nitrogen form on in vitro organogenesis on the field horsetail or common horsetail. So this is a common weed that is all over the world, and definitely early successional. If you want to jump into the nitty gritty of this study, right there are the creators of the study, and you should be able to find it with those keywords. So what they did is they grabbed ammonium and nitrates, and they did a study with the common horsetail to see if they fed it one or the other, how does it perform in its whole journey and life cycle? What is the yield of feeding the horsetail what is the yield? What is the production when they feed a horsetail its preferred food or not preferred food? And so here, the top line is NH4, ammonium, and the bottom line is NO3, nitrates. And so this is in the formation of vegetative shoots in the horsetail. And so they fed the grouping in the above line ammonium and the grouping in the below line NH3. So if you look, we're down in 2 and 1 with NH4, and we're up in 3, almost a 4 with the NO3, with the vegetative shoots. Now jump into the other ways these plants grow. So the first on the left here, the can you guys see my cursor? Sorry, I'm asking. Just tell me in chat if you can see my cursor. I don't know if you can.
I don't think we can too, okay, Chris.
No problem. So the rhizome formation, or the kind of root structure formation in the common horsetail there, as they fed it ammonium, which is that top line on the left, that is zero growth. That's nothing. So by feeding these common horsetail the organic N, this other form of nitrogen, they had zero production. So in natural farming, we've talked about seeing a decrease in weed abundance after 3 years in outdoor living soil, that the weeds change. We stop seeing those early successional weeds, and in less time, in one application, in 3 months, you can see a total change. But why? Excuse me. Why are weeds struggling in these highly diverse or highly fungal growing environments versus highly bacterial, and depends on what's planted, etc. That's not part of the study, but again, we're extrapolating information on our crop, which isn't necessarily studied this thoroughly on these topics yet. So here, zero production in their rhizome, in vitro rhizome, and with nitrates, we got decent growth, normal horsetail production on that bottom line on the left-hand side, figure 3. Going over to figure 4, we have tuber production. They have kind of, they can grow along and pop up in a new place and really multiply. And with ammonium, there's die-off. There's dead stop in production of this early successional weed, where with nitrates, we have a ton of growth, normal growth. Crazy, right? So, extrapolated out, hypothesis, but again, a little bit on how our soil food web, or the engine of the microbial community, diverse microbial community of our soil, how might that be affecting our yields, the plant stresses that show up, and how resilient plants are to plant stresses. Because things happen on our farm, right? Our plants get stressed. We forget this, or this happens, or an environmental change happens. And what we want is resilient plants. The less stressors we have in our growing environment, the more resilient a plant is to a stressor that comes in. Whereas if we built up several stresses that the plant's dealing with, now another stressor comes in, and that plant becomes very susceptible to decreased production, to germs, to things. I say germ kind of to use a term other than just microbes, more things that make a plant sick. So this study, again, you can dig into it. You can look at how they did it. It's published and there for all to see. But my hypothesis is, or what I expect on what I'm based on what I've seen, is that that cannabis prefers a organic N, when possible, an ammonium, and that it will do better when that nitrogen cycle is being inhibited from high amounts of nitrates and nitrites. The nitrates are food for our weeds. So just how much can fungal life affect nutrient uptake in crops? Here is a study, I think this is the one out of Spain, where they analyzed, I think they're olive, sorry, it's not, yeah, there it is. Our buscular mycorrhizal fungi increased growth, nutrient uptake, and tolerance to salinity in olive trees under nursery conditions. So high salinity is a stressor in environments. Some people deal with that as a result of formal growing practices or brackish water, or just build up over time and growing in the same space. And so the study looked at 3 types of mycorrhizal fungi introduced to growing environments and nursery conditions. This study I love because the replications, the amount of trees that they studied per group was massive. This was a huge study as far as actual labor and getting a big sampling of each of their trials. But what this is saying is that in stressor environments or poor soil quality, the presence of fungal life caused, well, you will see. That's a little picture of the undergrowth at this farm here in Colorado. This we did not inoculate with mushrooms. This is just indigenous microbials put down, and we get tons of different microbes. All the crew there at the farm are like, we're going to get into mushroom identification so we can figure out which one of these 100 microbes are edible. And it's like, okay, I'm not going to identify any of them for you because I don't want to be responsible for who gets sick from some strange mushrooms. But it is beautiful to watch and a lot of fun when we get these big macro fungi coming up and showing us that they're doing work down there in the soil. All right, jumping into the study, shoot and root dry weight. So if you look on that first line of numbers or first set of numbers, it's they did not add extra salinity. They did not add extra salt. And in the second set, right there in the first group of lines, table 2, we see they did add extra salt. So you're looking at a series of data points where how much shoot growth, how much root growth did all of these exactly the same trees, these nursery size olive trees, how much growth did they have in the different tests. So we have a control, which is just, soil and water. And then we have 3 different fungi, mycorrhizal fungi, introduced into those environments. And if you look across the line, that control there with salinity present, the shoot growth was 66.98. And that's centimeters, I think. Sorry, I tried to take down the study. It's massive, and I tried to get some points to help you understand the kind of outcomes or the implications, rather than try and go through the entire study because it's kind of a mouthful. But you look at almost a 300%, 400% growth with the first set of fungi present in that exact same environment. So only change between those two highly very over salted growing media environments. The only change is the addition of a fungi. Okay, so we have 206 versus 66 as far as root growth or shoot growth, and even in the environment without dry weight and centimeters, and then down with no addition of salts, you still have a massive increase, like a 300%, 200% increase in growth just by having fungi present. So if you're only using LAB, lactic acid bacteria, or EM, or bokashi for growing, and you're limited down in mostly bacteria, you might not be benefiting from this potential growth in your cannabis production as a result of just not having that thing that the cannabis likes to partner with in their root zone. So going down and through a couple more tables here, we see it repeated in different aspects of the trial. Going down to table 3, look at the difference. That first line is a control, no fungi, and then the next 3 lines are fungi present. And Mosae, I don't know that I'm saying that right. I am not good at Latin. Sorry. That introduction there, Efren can correct me on pronunciation. Thank you, Efren. The production increase, both in high salinity and low salinity environments, is pretty drastic. The difference that we're seeing just by one species of fungi being present in those root zones. Again, what could this mean on your farm if you tailored or catered to your crop's preference in kind of microbial communities in their growing zone? Again, this is not a cannabis study, this is a tree study, but I think that even especially nursery trees are probably the closer thing as we look at scientific studies to what our cannabis plants prefer, in my opinion, than really anything else that we can study. I think trees are the closest. Some shrubs like a blueberry or some of these things might be a great study as well. But I really like and think that we're closest as we're looking at how some of these trees grow. Even though a tree might prefer, 1,000 to 1 or 200 to 1, where cannabis might prefer 30 to 100 to 1, I think we're really going to see. We already have. We have continually seen those similarities, and that's why I'm talking about it. But as we continue in this delving into the science of it and doing peer review studies on how cannabis eats, I expect we're going to be far closer to a tree than a tomato plant. It goes on. We don't have to go through all these lines, but it was very conclusive that one, the environment or stressor was almost a non-issue. You see very little change in production when they put it into high salinity environment if the fungi are present. Basically, the fungi is buffering your plant roots from dealing with the environmental stresses of the soil. And that relationship is giving your plant a resilience to continue to produce at optimum levels, like if there was not high salinity present in the root zone versus if they don't have fungi. So that fungi is your helping with that yield, even in poor growing conditions. And what does that mean for you as a cannabis farmer? Could this have implication? I think absolutely. very much. This is just a quick view at what we're doing as we're putting on IMO 4, how fungal it is, and how close we're looking at the fungal to bacterial ratio as we apply inoculum. And so there is an art to creating good IMO 4, which is that fourth stage. One being that moment that we're collecting on a media, collecting the diversity of microbes from the forest or grasslands or long established microbial communities in nature, and moving it in, and then how well do we maintain that fungal dominance as we grow it out and apply it to our farmland. So this is just a snapshot at kind of what you're aiming at, and I would say a bit more fungal than your typical compost. All right, so this was actually some pictures I took yesterday, and we'll talk about other things we see, but the point of this slide is to say how vast is the microbial world that exists in a regenerative agriculture or living soil environment, and how can we boost or increase that diversity so we're benefiting from the massively diverse microbial communities, the very nuanced and different and interconnected microbes in soil and nature, and how can we use that or partner with that as farmers to grow the best crops with resilience, with bug resilience and disease resilience, as well as production and yield, because there are incredible nutrient cycling potentials in our root zone. And so what we're looking at here is a fungi that, oh my gosh, I didn't write the name down. It's nematophagous fungi, basically a fungi that is a predator to a nematode. So right here on the right, there's a bunch of different kinds of nematophagous fungi. This happens to be one that creates nooses. Think of it like a lasso. Those circles there are three cells or containers that in a split second, in a moment, that fungi, when it senses a nematode's head has passed through that little noose, that fungi can throw all kinds of cytoplasm into those nooses and create a pinch point and trap and capture a nematode. Then as that nematode dies, the fungi pierces its skin, grows its hypha into its body, lays and reproduces or sporulates and continues its life cycle. Bonkers, right? This is happening not because we inoculated with a bunch of nematophagous fungi or a bunch of nematodes, but this is happening because we took nature's diversity and placed it in our farmland, in our growing environment. In this case, 5 foot wide by 4 foot deep cedar beds full of soil, basically, and let it become established by tending to it, keeping the water right, the environment right, etc. And so on the left there, you see I believe a bacterial feeder fungi. Those big lips on the front has been colonized or killed, it seems, by a fungi. I mean, I can't say empirically that nematophagous fungi was responsible for killing that nematode. But interesting, these were on the exact same slide in the exact same water droplet, and we looked at both of them at the same time, or as we're scrolling around on one water droplet. So this is happening in our soil, and we're just finding out about it because our soil has diverse microbial communities and is cycling nutrients well. And that diversity, interconnectivity, I can't begin to understand how this normal thing that happens in soil is positively or helping with the overall diverse nutrient cycling of our plants. But I do know that a nematode, just like us, when it decomposes and breaks down in your soil, is water and nitrogen. We're getting that little thing on the left there is fertilizer as it breaks down and decomposes, just like if you were making fertilizer out of fish. Does anybody use hydrolysate or fermented fish amino acids, or what's the other form of fish? Hydrolysate and what? So the impact of the indigenous microorganisms.
Are you talking about emulsion?
Thank you, emulsion. I appreciate it. It fell out of my head.
Yes, sir.
Yeah. So what are those? Those are the decomposing bodies, or the bodies of organisms that are full of nitrogen and water, happening to be food for our plants. That happens on a micro scale, on a massive, it's smaller organisms, but massive scale, in soil that's alive all the time. That nematodes growing off of bacteria, and then dying and adding fertilizer back into your system. So we're not seeing this any lack of nitrogen in these living systems based on plant growth and yield, even though we're adding homeopathic amounts of nitrogen in comparison. I found the same thing in our orchard. When that soil was cycling, we had far less need for nitrogen. The community of macadamia nut farmers, as we first started talking about it, some incredible people that I learned a lot from as a kid, walking around with Dad and, kicking tires in the trees and talking about what the trees need and what some people are doing. Those same experts came out when we started playing with a 144-acre trial of natural farming, and they're like, you can't grow macadamia nut trees with that little nitrogen, because we're like at 2% of the recommended nitrogen, in a year. And then they came out and they said, oh, your trees look as good as any in the world. And then the head of the International Macadamia Nut Society said, hey, I think you should be on our research and development board for the International Macadamia Nut Society. So now you get to speak once a year, other than COVID, which sucked, but I get to speak once a year to peers, a room full of people a lot smarter than me, all PhDs, and I get to talk about, our project on our 750-acre farm and the implications of microbes on yield, where we're on an island. Our cost of inputs to ship in are so high. To be able to drop those input costs while maintaining yield has been a huge bottom line effector for us as a farm, as a community. We're really community oriented in our farm and love our team. And we've had a lot of benefit from just kind of saying, we don't know all that's happening in soil, but we're going to tend to the microbial community, the indigenous microbial community. And indigenous microbes tend to not have a fall off. They self-perpetuate in your systems, even if you're indoors, versus often what I'm finding with, or kind of we as a scientific community are finding with bugs in a jug, lab grown from a different place, is that there's about a 6-month fall off. They're wonderful for helping with production and yield. I'm not poo-pooing, all these wonderful products, and you need to reapply them. And there aren't creating lasting communities in the same way, per our observation. And so we're talking about indigenous microorganisms because of the unique and amazing ability for them to self-perpetuate and give us these benefits that we might be by and large unaware of. Like until I scoped this, I didn't know we had, these warrior fungi killing nematodes and turning them into fertilizer and sporulating inside of them. But that's what happens in nature, and that's one tiny little part of the interconnectivity that's going on there. Fun to look at, fun to watch. There was a ton of nematodes too, and good to watch. But As I said, I'd really like to move into questions a little early. I probably will, apology or just a heads up, I probably will do a bit of rabbit trailing off of questions, because I think sometimes deeper answers can really help with the understanding. But let's jump into what are some of you guys' questions or thoughts on some of these concepts, indigenous microorganism, and how they affect you.
All right, thanks, Chris. We've had a lot of questions piling up here, so we will jump right into them. So let's see. First question, somebody was asking, are there endophytes in IMO?
Yes. I don't have quantitative numbers. It hasn't been peer review studied, but we're seeing them.
Yeah, that's not too surprising. Let's see. Have you seen anyone have success with chipping up cannabis stalks to use as a carbon source for making IMO?
No, don't consider your cannabis stalks as a carbon source. Consider them as a green material like grass. They are not as woody as all that. So, no, you can include cannabis biomass in composting for sure. And I think the way to use that in collaboration or in conjunction with IMO production is to produce IMO using a good woody material as your carbon source with your current carbohydrate or grains. Again, grains, you want to have them higher in fat and protein content when making IMO. So oats are great, teff, rice bran, and many other things have a higher fat protein profile, which are fungal foods. And so do that. But if you have a finished IMO4, then getting all that nice green biomass, maybe bringing in a little bit of high nitrogen, very small. So something like 50% cannabis waste, 20% IMO4, and then filling the rest in with some carbon and a tiny bit of nitrogen, no more than 10% high nitrogen, and composting it with either actively turning. If you want to do it static, then you would balance your green, your cannabis biomass with high carbon material like wood chips, and then inoculate it with IMO4, 10 to 20%. And then you could turn that and let it sit, keep the moisture down. If you're going to do a static pile, you don't need a ton of moisture. There's a ton of moisture in your cannabis biomass, even though it seems kind of dry. And then you can let that sit, and it should process quite well over time. Come back to it a year later and mine underneath it. That's not a microbial inoculum because you're not being so careful on exactly how you're tending to the microbes, but it can be a very low effort, high quality compost that you produce. Those ratios are flexible based on what you have. Lanningham talks a ton about good composting ratios and stuff, but no, as a ingredient for that IMO 1 through 4 process, I don't think cannabis is a good addition.
Great. Actually, along those lines of static composting, Chris, are you familiar with Dr. Christine Jones and her work?
I have heard the name. I haven't studied her work.
Okay. Yeah, she did. I actually took a workshop with her last summer where she taught a static composting method. It was really interesting. I encourage people to check that out.
Awesome.
And sounded, yeah, it's kind of along the lines of what you were talking about there. Another question, you were saying that the beds you're currently working on are sand, silt, and clay, mix rather than like a soilless mix. So this, now I'm thinking maybe this is a better question for Leighton in his session, because I know he does this all the time, but do you have any tips on sourcing those materials for building a topsoil?
It's really simple. Yeah, so that isn't so much a topsoil. Leighton is a great one to ask about this. This is really the part he played in this project. It is something I've gotten to study and play with and continue with. But yeah, he's a great one to ask. But we are using river rock from the area, so just river rock, and then sand, silt, and clay from the area, and you just want it to be as least processed as possible, a place where somebody has availability to mining some of this. And so you just want, and what it is there's just so much in there. Everybody's like, oh, silica, I need silica for plants. If you have a functioning soil food web or microbial community, and you have sand present, they're going to mine the silica for you. Over time, you will get that. And so this is just to put those mineable materials in there as the soil matures. Those will begin and continually get mined and break down and become plant available. So this is, we've built what we estimate are 10-year beds, beds that don't need to be changed for a long period of time. So we avoid a huge quantity we avoid just dumping a bunch of nutrients in there and keep it pretty simple, because we're really looking for those materials to interplay and mature over time. We may be adding a little bit of material periodically, just to as things decompose and shrink, we'll re-up. But yeah, sand, silt, and clay isn't complicated. So don't overcomplicate exactly what that is. The main thing, you just want to stay as least toxic as possible. You want to make sure that there's not weird stuff in there that you'll be surprised by, like industrial waste or something.
Got it. And actually, I just remembered Leighton talks a little bit about this in a Shaping Fire podcast too. So if you guys go look that up. Let's see. So a question about using eggshells for making calcium. Have you ever seen a difference between like your backyard chicken eggshells versus store-bought eggshells?
I have not studied that. I think I would suspect that a really healthy chicken that has enough calcium in its diet, homegrown or free range, and it's eating bugs and dirt and plants, is going to have a much healthier shell. They're a lot harder to break when I try and crack them versus a store-bought egg. So I would suspect you have a lot better nutrient quality and quantity in a well-raised chicken that's healthy versus a bit anemic, production for chicken. That's not to say that's true across the board. It could vary back and forth. However, a better form of calcium for water-soluble calcium is coral sand, calcium carbonate. And you don't have to pre-cook it. You don't have to do anything. You just put some coral sand, 1 part coral sand, 10 parts, 10 parts rice vinegar if you have it. You can use other vinegars, just not white vinegar. But coral sand's super easy. That's what I use for a massive scale on our farm.
Oh, wow. Awesome. A question here. I don't know that you mentioned this in your talk, but relevant to KNF is someone was talking about why use 3 different types of fruit in FFJ if you only use 1 type of plant in FPJ?
Good question. Sorry, I tried to inhale bacon. It didn't work for me.
That's never worked for me either.
FFJ has a very, very different enzymatic profile than FPJ. And so if you're just starting out with natural farming, I encourage you to forget that FFJ exists and stop using it altogether. If you're a 1 or 2 year veteran, I think FFJ can have a very limited function on your farm. But if you are starting out with natural farming, wait until you understand the function of FFJ as a very small and limited use tool before you go and make a bunch of it and try and use it in cannabis. It is a common mistake in early people starting out in natural farming to think FFJ is super cool, all these wonderful flavors, they're going to be so great for my cannabis. It's not really how it works. So if you're not been doing natural farming for, where you really understand how your plants react to these things over a 1 to 2 year period, I would forget about FFJ for now. To answer the question, fermented fruit juice, FFJ, which uses ripe fruit. The reason we bring in several is because we're capitalizing or we're embracing that ripening enzymatic profile that happens in ripe fruit for promoting that finishing off period in our crop. However, that said, I have not seen a great deal of evidence in use in cannabis that there's really any benefit to using FFJ. And so it is my recommendation that as cannabis farmers, you stay far away from fermented fruit juice unless we find some peer review study or some side-by-side that somebody does that shows that it has a function. I think it is very, very limited. We did trial some of that kind of finishing material, FFJ, in some of our crop this cycle here, and we found very inconsequential, or we found no difference, no benefit to using that finishing. So is my recommendation for the cannabis community to forget that FFJ exists and focus on FPJ.
Awesome. Let's see. I'm just going to say there was a question about soil horizons. I encourage you to save that for tomorrow when Leighton and Chip are talking about soil horizons. So let's talk about this fungal to bacterial ratio, because you said that, and then we had Elaine Ingham on here last time saying that she thought 0.8 to 1 was the ideal fungal to bacterial ratio for cannabis. Honestly, I'm not sure where she came up with that. But I'm just wondering if it sounded like what you were saying was we're not entirely sure, but we're making some guesses. Is that right?
We're not entirely sure, but in the systems where we know about where we're at on fungal to bacterial ratio, we're seeing a huge, or we're seeing an increase in those highly fungal environments. We're seeing a significant increase. We're in addition to that, we're seeing greater resistance to disease, and even what I suspect is thicker cell walls that give us bug resistance. Though cannabis is always going to be susceptible to bugs to some degree, and we have solutions for that through genome tools. We're seeing that in the systems where the fungi has been established and is in greater quantity, the plants are doing better. And that's a lot of it's anecdotal. It's not like we've done some university study in collaboration, but I do get to talk to a lot of the best, a lot of people that are doing natural farming well and having success. We've dialogued in the U.S. anyway, and some internationally. And so, that comes out of what we've seen. And again, I kind of say all of that very carefully, as these are hypothesis and observations over time, but not anything with comprehensive data. So this is my best that I can give you at this point, and totally willing to be corrected or find new information that leads us to a different number.
Mm-hmm. All right. And one more thing to bring up about Dr. Ingham, and not to try and cause any controversy here, but I think it's good to get a different perspectives. She came to our last session, and I think somebody directly asked her about the whole aerobic versus anaerobic thing, and would you put anaerobic ferments into your garden? And she, was kind of like, no, I don't think that's a good idea. I'm sure you've responded to this before, but just wanted to get your comments on that.
Yeah, me and Dr. Elaine have sat down and really talked about a lot of this at length. And I think that was since. Yeah, I really, really respect Dr. Elaine Ingham. When I was starting out in 2011, she did a seminar in my tiny small town in North Kohala. And it was right about the time that I needed to start quantifying my microbial communities and some of these things I was doing for use on our farm. And she taught me how to use the microscope. I bought a microscope that same day, and it was huge. And so I think she has she's one of our OGs in regenerative agriculture, and has done incredible work and gotten incredible pushback from the broader scientific community, because this is a new frontier. We're learning as a community together. We're growing and learning way more than we've ever known about microbes in community and their effects on plant growth and yield. So I suspect, especially with cannabis being something that's kind of lacked in great studies through collaboration with universities, I suspect we'll get you some better numbers and answers that are empirical evidence and data-driven over time. And that's one of the things I'm most excited about, is collaborating with universities. I'm getting to do some of that. And so I look forward to more. And if you see opportunities in your spheres of collaborative studies, I would love to be involved. And I offer that. You can email me right down there. But what we're doing with ferments needs to be understood to be of homeopathic quantities. So we're not dumping anaerobic ferments into our soil and hoping it all shakes out. We're putting, 1 to 1,000 in water. So in a liter of water, we got 1 ml, of some of these things. You can barely taste it if you drink it. The effects of that are more beneficial biochemicals, auxins, plant growth hormones. These are really what we're doing with those ferments, and some of them are aerobic and fungally fermented, like the fish. And so it's, I think, understanding when we say we're putting on ferments, that it is, this small part in the 20% after the 80% that's IMO. It is very effective. And we're not messing with our microbial balance, either on the leaf or the soil, according to our observation or the kind of community observation. So I get it. I get the reticence. We start talking about vinegar and alcohol and ferments, and everybody's like, "Oh no," But I have sat down with Elaine, and we've talked about that. And she asked if my farm could be on her website and if she could come out and study what we're doing out there in Hawaii. So we love each other and get to share. We shared some papers. Last time I went to China, I asked her for a paper that she had access to that I couldn't find, and she asked me for a couple of that I had. And it's we're part of a community together. It's not we're not fighting, and there's a lot to learn as a scientific community. So I think I'm not worried about those questions or tensions or that, and happy to field them and happy to sit down and talk with them. With Elaine anytime. We have gotten to speak at the same conferences and hash it out, and we don't have issue. So I get those questions, and dumping alcohol on your soil would turn your roots into liquid and kill most everything. Nobody's doing that in natural farming, and if you are, I'm sorry that you're missing it.
That's right. Awesome. Thank you for clarifying that, Chris. I think that was probably really helpful to a lot of people. So, a question about how do you judge the quality of your IMO 1 collection?
That's a great question. That's probably one of the more nuanced and harder things. I'll give you a couple points that are really important. If at any point you have slimy or gooey centers or places in your IMO collection, that's a total failure. Those are you've begun to rot or putrefy. And that won't ever make great IMO3. It's not a good inoculum, and I'm sorry, but just throw it away. One of the best ways to make IMO1 successful is get yourself 8 collection boxes, 6 or 8, make them all at once, put them out as you go out, and collect them all as you come in with the expectation that you'll have a couple that go really well. I feel really bad for the people that put one out, it fails, and they have to go 8 times over 8 weeks to get a success, whereas I want a success next week, and so I'm going to put out 8 and get a few successes, and it's okay to fail. This is not something where we're controlling all the environmental factors. Another thing is there's a bit of a misnomer that it has to be perfectly white to be a proper collection, and that comes from South Korea, where a large portion of natural farming happens in rice farming. And in rice farming, they're putting out their collections in these places with their rice. And they're one of the main microbes they're focused on is Bacillus subtilis, which is present in like rice straw. And it's great for producing rice if you're a rice farmer, but it blooms perfectly white. But what we want when we go up into this wild, forest preserve in Colorado or out into the jungle in Hawaii is we want all those different fungi present there to be as represented as possible, or as they exist in the diversity they've struck in that environment. And some of those fungi, to the naked eye, have colors. And that's okay. What we'd want to avoid is a all orange collection of vomit mold, or a solid green collection of, help me out. What's the green? Trichoderma. Thank you. We don't want to see these one-dimensional collections where something got in, or maybe it was too wet, or we left it too long, and there was balance maybe when it started fermenting, but then because we weren't there on the day it was ready, something ran away and cannibalized all the stuff that bloomed and died off. So you just want to be aware that light white, light gray, and even sporulating black heads. People are like, "Oh, it's dark gray. That's bad." Well, if you take a white bloom of fungal growth on something and you touch it, and you just press on it a little bit and take it from expanded and fluffy, and you just kind of push it together so it's denser, it turns dark gray and black. Because what we're seeing in that white is we're seeing through that organism. We're seeing layers of it as a fluffy thing. But as it sporulates, like a fungi sporulating head, you can't see through that anymore because of the density, and you see dark gray or black. That doesn't mean that you have black mold. And black mold isn't even really something that we're worried about in nature. We're worried about black mold where Aspergillus fungi got into a house where the drywall got wet, and it's consuming the inside of drywall or decomposing it, then sporulating and going aerosol, and we're breathing basically drywall toxins, which is some of the most toxic stuff in the world. All this to rabbit hole to say some colors are great. We do want to see some of that white bloom, and more importantly, we want to see no no gooey centers, no liquefied portions of rice, and really we want the rice to be almost held together by fungal hypha. We want to see and be able to break apart and see all those striations and strands. And so, yeah, I definitely need to teach more online about IMO 1, but in person, we really get into it when we do these in-person classes and people have a good understanding. So I'm sorry that there's not more information available to you yet. But yeah, I hope that helps to answer that question.
I think that's great. That's a good start for people. So just keeping on the theme here of IMO and quality, what about judging the success of IMO 3? Somebody asked that too.
IMO 3, if you really don't know at all, you can spend $100 on a Earth Ford trial or some of these other microbe counting labs where they'll analyze your fungal to bacterial ratio and your hyphal diameter and some of the other microarthropod or protozoa, etc. You can do that. I showed a slide there which represents this is just an IMO 3 sent away. And when I was learning the microscope and first starting out, what I did to kind of calibrate my eye is I would send away duplicate samples. So I was working, our macadamia business is a multi-million-dollar farm. So we had some money to spend on, dual. But I would take the same sample and I'd put it into 2 bags and I'd send it away with different names. So that I could get, because there is some variance in counting, and so I would get multiple analysis of the exact or duplicate analysis of the exact same sample, so that I knew where the variance was, or at least a little better idea of the variance. But before I sent that away, I would look at the same sample under a microscope and give my thoughts. Basically take that sheet that they give you and make it all blank and give my kind of count. And I would say, okay, I'm trying to count and approximate the biomass of fungi versus the biomass of bacteria. How much of these am I seeing? I'd write it down, send away my samples, get it back and see how close am I seeing what they're spending all that time counting. I think counting is a wonderful way to do it. They count per field. And it is the scientific way to do it currently. But, as a farmer, I would get a little flack managing the farm if I spent too many days in front of the microscope. So I tried to find a balance of analysis that was accurate while being able to move forward and use that data to affect or change my protocols if need be. And a microscope is a good tool to analyze your quality in IMO 3, or a send-away test to a lab that'll analyze your microbes. But not everybody has or can have access to those tools. And so a great tool, and better if you've smelt right and wrong before at a class or something, because your nose, your olfactory sits on your cerebral cortex and has a memory all of its own. And so your nose will remember right and wrong if you've ever experienced it. But a great general rule is keep it under 120 degrees. This is plant-based compost, so we don't need to get high heat. And don't let it go higher at night or anything like that. A great tool for that is an Inkbird thermometer. It's a barbecue thermometer. One of my friends found it. I'm like, if only we could get alerts to our phone. And Wendy Kornberg said, I found this thermometer. Now we all use it and love it. But what's great is it'll alert you at 10 o'clock at night that you're getting a little high. And so instead of that going all night too high, you can go down, turn your pile, or if your pile's at your house and you just forgot to check it and your alarm's going off on your phone, really easy to use app. I think they're $50 to $100 for the thermometer, and I love those. So keeping it under 120 degrees. 122 is, I would say, the upper threshold, because what happens if you stay really hot is you'll lose a lot of your diversity. All that stuff we're kind of fighting for maximum diversity from our indigenous environment will not, it might not go away. It might still be there, your initial inoculum in there, but it won't multiply and increase in density and quantity. So what then will happen is you'll get kind of actinobacteria running away with the space, especially once you break 125 or get up to 130. And looking under a microscope, you'd see solid monofilament wire, which is those bodies of actinobacteria. And it will smell a lot like ammonia. And what that is the nitrogen present in all those microbial bodies, and the grains are off-gassing into atmospheric nitrogen. And you're losing a lot of the nitrogen quality of your IMO, and then also a lot of the diversity. And so if that happens with our pile, we count that as poor quality. We don't use it as IMO. It's great for, adding to compost or etc., but don't consider it a high quality additive for diverse microbial communities. So that nose. It's one of the most effective scientific tools known to mankind, is the human olfactory system.
All right. Awesome. Someone was just wondering, can you collect IMO during winter? And I assume they're talking about winter and cold climates.
Absolutely. That's probably because I was talking about it on Instagram. We just did a collection in winter. We went up to 7,000 feet in Colorado, and I walked through 4 feet of snow to get under some trees and dug away snow, chipped away, an inch of ice, and underneath was all these beautiful fungal hyphae and soil microbes. They're present, visible, doing their thing, probably very dormant, but taking that down, a bucket of that down, and then laying some of it into a cardboard box, putting your rice collection in with it, and covering it and letting some of those fungal hyphae go right in on the rice, just a few of them without any organic matter on it, tuck them in under the paper towel, and then leave that for 5 to 7 days, depending on your temperature in there. I'd say generally about 5 to 7 days at 70 degrees. If you're colder, it's going to take longer. At 40 degrees and lower, you're not going to get an IMO1 collection ever. There's too little activity to really move in and inoculate your rice. So believe it or not, I think IMO1 is an incredible tool in having learned from Lane and Master Cho. I would say that IMO2 is the beauty of natural farming, or the special thing for a farmer to have their own indigenous inoculum that's shelf stable. People talk about JMS. They're excited about JMS. JMS is great, but every time you want to make JMS, which is Jadam Microbial Solution, it's really simple. I teach it. I love it, especially for third world. If I'm only there for a couple of days, I can teach them something highly valuable that they can use for generations. It's great, but I'm ripping out soil and microbes every time I want to make it. I'm ripping them out. Whereas I can go to I can place a box in a wild environment and then take nothing away that I didn't come with. And I leave nothing behind that I came with. And then if that inoculum, that IMO1 goes well, I get it to go dormant, or my fungi sporulate, things cyst in this low water conditions that the sugar, the osmotic pressure of the sugar creates. And so now I have a shelf-stable inoculum from my indigenous environment, and I can pull from or scoop out and get an inoculum from anytime I want. And right now on the shelf at this farm I'm at now, I think we have close to 15 or 20 different collections from different seasons, spring, summer, fall, and now winter, in Colorado, and from different environments, high and low elevation, in environments that look like this and environments that look different. Those are all on my shelf, mix them together to make my IMO 3. And now I have an inoculation of diverse fungi, and that's an incredible tool for a farmer to have this self-made inoculum that's shelf stable. Farmers are busy. I don't have to run out to the woods and harvest these rich environments every time I want to make a microbial solution. I have it right there on my shelf, ready to go in my farmland. And so I think people like JMS because it's easy. And I would say it's much more limited in fungal diversity. You're getting a lot more bacteria and yeast because it is not really an aerobic ferment. It's not really something. Whereas in IMO production, it's all about aerobic production of microbes or multiplication of microbes. So, so winter collection, I bring that material down, I set it up in a warmer space, and basically do a collection with a little bit of artificial environment creation so that those microbes can brew. Actually, I have an IMO 1 going on the farm right now that we'll probably check on Monday for winter collection. All right. And it's cold outside.
All right. Awesome. So someone is asking just about, saying they're kind of confused about getting IMO inoculations to stick around in an engineered soil versus like a native soil.
So it really is a good concern. I think that in potting mix alone, you're going to have a harder time self-perpetuating microbes because of the nature of the environment they're living in. That doesn't mean you can't do it. One of the ways we talk about doing that for people that they got what they got. They got their beds full of what they got them full with, and they need to try and farm as best they can. They want to move to living soil, but they're kind of reverse engineering living soil because they started on an organic protocol or something. Liquid IMO, which is an actively aerated compost tea with natural farming inputs, I came up with that mix of inputs through just years of trial and error, looking at it under a microscope as I brewed it out in a water column. And that is a way to continue to re-up inexpensively rather than have to buy a product all the time. And in that, you're going to have tons of lactic acid bacteria, great indigenous yeasts. And if you have a substrate for them to grow on in the water column, like a compost bag for composting tea that's big enough for fungal spores and hypha to go through back and forth, or theoretically, the fungi will grow on that bag out of that substrate that you put in there, that IMO4 you put inside, and it'll get covered in fungal hypha, and then those will break off into your water column. Some will produce in the water column, but really I found that if you have a very active bubbling or aeration system, the fungi have a hard time growing in the mechanical movement of that water more than anything. It's not that they can't grow in that air environment or that moisture environment or those nutrients. It's literally that if they're constantly being broken apart by the movement of that water, you're going to have limited production. And I found that the bag itself actually acts as a holder for them to grow on in diversity and in community, and then they break off into your water column. That is a way for somebody in kind of a fabricated soil that maybe isn't really as kind of long-lasting or kind of inert or natural as what we're talking about here. But, really possible to get a lot of the benefit as long as you're kind of maintaining that microbial community. But you will have a harder time in like a peat moss or cocoa to maintain a diverse microbial community. and it will require reestablishment probably.
All right. Awesome. So we've got about 10 minutes left. I've got 3 more kind of key questions to get in here, I think.
Okay.
So one person was asking, commenting just about, how their goal when they're growing cannabis is more about quality and cannabinoids rather than yields. And so, maybe can you just talk a little bit about...
Can you repeat the question, Daniel?
Yeah. It's kind of a hybrid comment question, but I guess the question is, should we be using different techniques if we're aiming for quality cannabis and higher cannabinoids rather than yield?
Absolutely. And we're back to talking about microbial diversity and inert minerals in your soil. You ever had I'm going to ask a question that you can't answer, but you ever had a strawberry? I'll ask you, Daniel. You ever had a strawberry from Costco that looked absolutely beautiful? Not Costco, anywhere. It looked absolutely beautiful, but tasted just like water.
Oh, yeah.
Yeah. Well, those, hydroponic strawberries or strawberries grown in a highly commercial salt system, they're given everything they need to grow big and beautiful and red, but not everything they need to produce complex biochemicals or complex sugars that we associate with flavor and fragrance. So the living soil. Now, at the same time, you ever gone out and you found a little wild strawberry and it was way too small, but you popped it in your mouth and it was just like an explosion of flavor, right?
Yep.
What's the difference there is one has all the nitrogen and potassium and phosphorus it needs and a few minors, but really it obviously didn't have what it needed to produce real full flavors and complex sugars that we associate with flavors. And the same, I think, is true in cannabis. And we've, as a natural farming community, have talked about this a lot. And there's far smarter people than me to talk about this, far more educated cannabis growers. But I'll repeat some of what they're saying. And what I'm finding is that in living soil systems, it seems that cannabis is able to get on-demand access through those symbiotic relationships in their root zone and the nutrient cycling of microbes, where they're able to get these minor minerals as they need them so that they can put together and compile and create in their systems these complex biochemicals. But they're not going to get them with really just the pink and green. You can wrench on maybe THC, but to get that broad spectrum, there's 64 cannabinoids that they've identified potentially. That number's changing for me all the time, so I'm not sure if I'm up to the current number. Yeah. But any one cultivar could express any number of those biochemicals as its typical expression if given the ideal conditions. So say maybe one cultivar typically or has the potential to express 23 unique cannabinoids, but in a limited nutrient environment, it's only going to really show, 4 of those or 3 of those or 1 primarily, whatever. Whereas if they're getting diverse nutrition or complex nutrients available to them, they're able to bring together the building blocks to make those complex biochemicals or to express more fully those cannabinoids. Also reducing environmental stress and making those plants as healthy as possible also contribute to that highest quality. So I think I'm kind of I have a view on things and a way I do things, and it's natural farming. It's this protocol that we've developed for scale, but learned out of South Korea, and it's not the only way to work with the microbial communities of nature and cause them to be in our farmland for the benefit of growth and quality. But it is an elegant method. That's why I like it. It's concise enough that we can do it, but it's not so reductionary, that we're excluding all these wonderful natural phenomenon and dynamics that are at play to get that plant to be able to express its full potential expression. And so, you know, the conversation is that it's not just about THC, because that's the effect of that is a buzzy, high, sure, but that feeling, the euphoria or the happy, the giggles, and all those different things that can affect our body and mind in using this as medicine, those we get from really healthy and nutrient dense plants. So one more thing on that question, I think this is an important one. They did, this is anecdotal, but they did a bricks analysis on the cannabis cup submissions. Cannabis cup or emerald cup? Anyways, cannabis cup submissions.
I think I heard
you heard of that.
Yeah,
so they did a bricks analysis, and the top 10 nutrient dense cultivars that they had were the top 10 placers. So that association with nutrient density and what we associate with quality is there. There's precedence for that. So again, more to learn. I don't know. That's as much as I got for you on that one. But I think it's worth looking into. That's
perfect. All right, so we're going to get cut off in about 3 or 4 minutes here, Chris. But 2 more quick things here. Someone was asking about what's a good place to follow empirical research on KNF practices specifically, like who's doing this research. And I'm really curious about that too. And then, for somebody who's just getting started with KNF stuff, where should they go?
2 great questions. For somebody just getting started, I think starting to play with it without fear is the best way to go. Just start playing with it. I have 23 videos or something like that on YouTube. They're free. Make them. Play with it. I mean, really, though, that's the place to start, is doing it with your hands and experiencing the way things can go right and wrong. I have a website with some downloads, free downloads for PDFs, christrump.com. And that just gives you kind of, it's not comprehensive, but it gives you a basic supply list so you know what you need on your table to do these things. Because otherwise you're running around looking for that funnel that you didn't know you needed. But I think playing with it is the answer for that new person. And then as far as peer review studies, it's really limited. Natural farming landed really for the first time in the U.S. in 2008. Very little awareness of it, and in 2011 a group of us learned from Master Cho and Ju Young. That was really a launching point for the American natural farming movement, and that's really recent. So there have been some really horrible studies done where people that knew nothing about natural farming but were scientists and wanted something for their master's thesis, jumped on and screwed everything up, and then were like, this is what I found. And that's been kind of sad. But then there's been some great studies done, but it's pretty limited. The most studies that I know of are in Japan, in Japanese, and in Korea, in Korean and some in English. Kongguk University is a university that has a doctorate program in Seoul, South Korea for natural farming. So I advise their master's and doctorate thesis and have good relationship with the head of that school. And they're doing some great research, wonderful research. And so I don't know how accessible that is. And we will continue to talk about the research of this in online forums. And there are some research projects in motion, but there's not a lot. So I apologize. It's just too new. But I would love to work with universities, and I am currently. So as those research projects come to fruition, I'd love to share them with you. And if you know of some, share them with me, and I'll push them out so you guys can hear them.
Awesome. Well, Chris, this has been absolutely amazing. Thank you so much for joining us here.
Thanks for having me, man.
Yeah. And so Leighton Morrison is next up in an hour here. Just want to say to everybody, if you're having trouble finding the emails, the links, check your spam folder. Some people have been getting them going into the spam folder. You should get an email 15 minutes before the presentation reminding you about it with the link. So just check your email there, and we'll see you all in an hour.
See you guys. Thank you.
All right. Thanks again, Chris. See ya.